I'm just going to take you through a little bit about background to genetics and then talk a bit about the genetics specifically of tuberous sclerosis complex. So if we move on, Luke, to the next slide. So first of all, it's I think important for people to have a bit of an understanding of what a gene is. And forgive me if everybody knows this already, but I thought I'd just go through it. So a gene is a stretch of DNA, which is a chemical that contains a sequence or instruction that has a particular function, such as to make a specific protein. So we used to think that all genes made proteins. We now know that there are some things called non-coding genes, which do not make proteins. And actually, increasingly, we're discovering that some diseases are caused by genetic changes in these non-coding genes. And that's a really, really recent development, which is helping us to make diagnoses for conditions that people did not previously have a diagnosis. So I'm sure you'll all be familiar with DNA, which is this double helix that you can see over on the right-hand side of the screen. And that's made up of these chemicals called adenine, thymine, cytosine, guanine. And imagine that that's a bit like the alphabet. It's a word composed of just of those letters instead of the 26 that we have in our alphabet. And that's where mistakes can happen in terms of the spelling. So we have about 20,000 genes which occur in pairs, and they're packaged onto these structures called chromosomes. And we have 46 chromosomes again, which occur in pairs, and we get one of each chromosome from our mum and the other of each chromosome from our dad. And in the same way, we get one of each gene from our mum and the other gene from our dad. And of course, we have 46 chromosomes. 22 of the pairs are the same in men and women, and the 23rd pair are called the sex chromosomes because they determine our sex. And so women have two X chromosomes, men have an X and a Y. And for the purposes of tuberous sclerosis, they are irrelevant because tuberous sclerosis affects men and women equally, but quite differently. Um, so this is just an example to show you a little bit, as a sort of schematic to show what a chromosome looks like. And these are called a homologous pair just because they are the same. They're either chromosome 1, 2, 3, or 4. And the genes are arranged along the chromosome in the same order, pretty much wherever you are in the world. And I always make an analogy with imagining that our genes, which are, oh, collectively, we call the whole of the genes our genome. And you can see here, it's like, actually, no, sorry, I was on the previous slide still. The genes are arranged in a library effectively, so that the chromosomes are, imagine they're like a bookshelf, and on each of the bookshelves, you will have the genes, which are the books. But actually, overall, if you take all our genetic material called the genome, actually only 2% of the genome actually makes proteins. And it used to be, the rest used to be called the junk DNA. It's now called, because we know it is not junk, because of that, we now know a lot of disease is actually caused by changes within that junk DNA. We now call it the dark genome. And I went to a series of lectures last week about the dark genome. So if we move on to the next slide, please. So I talked about that alphabet of the C, A, G, and T being particularly important. Now, we're all different from one another, so we all have variation in our genes, and that's what makes us different from one another. However, within the genes itself, we there are those the genetic sequence is largely similar. When the human genome was sequenced back in 2000 for the first time, that became what we call the reference genome. But we very rapidly realized that that's only one version of the genome. So your genome varies according to your ethnicity in particular. And one of the problems we have is that the reference genome is largely based on Caucasian people. So we have a much better reference genome for people of Caucasian background than we do for people, say, of African background. And Africa, as people will know, the cradle of mankind, that we have more variation, genetic variation amongst the people from different African countries than we do in the rest of the world. So that's something that one needs to remedy. So some variation in genes or in the dark genome is completely normal. However, other variation causes disease, and we call that variation pathogenic variants. And the old terminology that we're sort of moving away from now is that's a mutation. So when a mutation actually causes a disease, a variation causes disease, we used to call that a mutation. We now use the terminology pathogenic, and sometimes it's actually hard for the people in the lab to determine whether a variant is actually pathogenic or completely benign. And so one of those things that might just be because you're of a different ethnic background. And when we have that type of variation, we call this a variant of uncertain significance. And so I tend to call that a VUS, but some people call that a VUS. And that's what challenges our scientists a lot of the time is trying to work out whether one of those variants is causative or not. And sometimes we are left in limbo, not certain whether that variant that we found is the cause of somebody's genetic disorder or whether it's an incidental finding and just a benign variant. So that's a bit about the background. So moving on to tuberous sclerosis specifically. So we know that there are two genes that are known to be associated with tuberous sclerosis complex. And these are genes, we all have these genes, and it's when we have a variation in the genes that people will develop tuberous sclerosis. So the first gene to be discovered, to be located was TSC1, and that sits on chromosome 9. And the second to be located with TSC2, which is on chromosome 16. However, the first one actually to be identified to work out actually what the sequence was TSC2, and that encodes a protein called tuberin. It was just called that by the scientists that found the gene, and TSC1 encodes a protein called hamartin. And you can see there that the pairs of TSC1 and TSC2 sitting on chromosome 9 and chromosome 16. And there you can see the one healthy copy of the gene, the green copy, and then the one that's got the variation on it indicated in yellow. And tuberous sclerosis follows what we call an autosomal dominant pattern of inheritance affecting boys and girls equally. But in about 2 out of 3 people with tuberous sclerosis, that genetic change arises by chance. So it arises in the egg or the sperm that makes that particular individual. And we call that de novo. So very often, if you see a geneticist or you genetic counselor, they may use this terminology. De novo just means by chance, arising as a new event. So variants in TSC2 are much more common. So we're more likely, if I meet somebody in clinic, it's more likely they're going to have a variant in TSC2. But if you have a variant in TSC1, it tends to cause milder disease and it's more likely to run it in families. But that's very much tends to. So I certainly have some patients who have TSC2 who are very mildly affected, and I have 1 or 2 who have TSC1 who are quite severely affected. So it's very hard as a clinician to predict when we have a genetic test result what the outcome for that particular individual is going to be, particularly if they're a young child, which is who we commonly see. So next slide, please, Luke. So this just explains a bit more about autosomal dominant inheritance. Autosomal means it is not sex-linked. Okay, so the chromosomes 1 to 22 are called the autosomes. And so any gene that sits on any of those will be referred to as an autosomal gene. So here we have 2 parents, mum and dad. Dad has a pink gene. So he has a genetic disorder, in this case tuberous sclerosis, but he has a white gene on the other copy of his chromosome. And mum doesn't have the condition, so she has two white copies. And when these are passed on in the sperm, or the egg, you have to reduce the number of chromosomes that you're passing on to a single one, because if the child got two copies from dad and two copies from mum, they would end up with four copies of that particular chromosome, and that is not good news, doesn't work. So they reduce the number of chromosomes that they're passing on. So each sperm just contains one of each copy of the chromosome, and each egg has the other copy. So when they come together, effectively you get the two copies of the chromosomes coming together to make the new baby. And so dad can either pass on the one with the pink gene or the one with the white gene. And so it's a 50-50 chance that he would pass on TS to his child. So see here, that's straight 50-50. And what mum passes on doesn't really matter because both of her copies are white genes. So I hope that's clear, and that's why it affects men and women equally, because it's an autosomal condition. So next slide, please. So, what do they do, these TSC1 and TSC2 genes? And in fact, it's the proteins, it's the hamartin and tuberin, which are really what are doing the work. So, hamartin and tuberin form a complex together. And actually, Luke, if you just press return again, it just indicates here on this schematic where TSC1 and TSC2 are sitting. Thank you. So TSC1 and TSC2 form this complex which sits in the middle of this sort of cascade, and you can see it's interacting with all these other proteins in this cascade. But the ultimate purpose really is that this complex controls how cells grow and divide. And they do that through this structure, which you can see called the mTORC1 complex, which includes mTOR. So you can see just below TSC2 and TSC1. There is a second mTORC complex called mTORC2, which you can see just to the left of the TSC1, TSC2 complex, which doesn't seem to be sort of quite so important. It's the mTORC1 complex that's important in the pathology of tuberous sclerosis. So essentially, they act as tumor suppressor genes, which means that this complex is controlling how cells grow and divide. So when you have a genetic change that affects how the complex forms or performs, then that means that tumor suppressor function is lost and cells start to grow out of control. That term autophagy down at the bottom, just to explain what that is, that's effectively where cells die. So they sort of auto combust, as it were, and that's just the term for cells dying off spontaneously. But as you will understand from our conversation sort of during tonight, is that the understanding of the biology of all of this has led directly to us being able to treat tuberous sclerosis. It was only by understanding what hamartin and tuberin did, the fact that they inhibited this mTORC complex, that we were then able to work out that by giving a drug that inhibited the mTOR complex, we could mimic the effect of that complex. And so that's what's led directly to treatment. And so alternative treatments are looking again at this pathway to see how might we influence that pathway in order to actually provide, even better treatment than we have already. So next slide, please, Luke. So when we do genetic testing in tuberous sclerosis, so if you've been to the clinic, you might have been offered a genetic test. We know that genetic testing is not perfect. That's the first thing. We know that first of all, that about 85% of individuals with a definite clinical diagnosis of TSC will have an identifiable, what we call a pathogenic or a likely pathogenic variant. And pathogenic means we're certain this is the cause. Likely pathogenic means we're greater than 90% certain that this is the cause, and that those variants can be either in TSC1 or TSC2. Of those 85%, and as Chris has already mentioned, about 5% will have a bit missing on chromosome 16, and that's called a deletion, and that removes both TSC2 and this gene polycystic kidney disease type 1, and means that people have both presentations. And you can see just from this schematic on the right that that's the chromosome 16, it's up at the top. If you miss that bit, you've got these two genes, TSC2 and PKD1, sitting right bang next to each other. And so if you just unfortunately have that bit missing, that means you have both genetic disorders. But generally speaking, uh, uh, there are no, well, there are a few common variants, but there are many, many different variants identified in many different families. So well over 1,000 different variants have been identified, and it's very common for us to find a completely new variant when we're testing people. And that's what makes it quite difficult for our scientists, because they're constantly finding new variants that then have to assess the effect. So that's why these variants of uncertain significance are really quite common and puzzle us. And then just a word about genetic testing. So we don't need genetic tests quite often to establish a diagnosis. And so we don't offer it to absolutely everybody. There is a bit of a move in the NHS, to be offering genetic testing to more people. But we do have to think also about our resources, because genetic testing is not cheap. It's several £100s to do a test. And so we need to work out, is it going to affect management of that particular individual? So first of all, is the diagnosis uncertain? Will it help to confirm a diagnosis? Or if this is a child and the parents are considering having further children, we should do testing because they might want to consider a prenatal test in a future pregnancy. Or could it be that a parent might be affected themselves? And indeed, we do find that we find a child affected, and then in retrospect, we find a parent to be affected. And so they need to be followed up themselves. And so each time we make a bit of a judgment about whether to offer genetic testing, we tend to err towards offering it rather than not offering it. But if we think actually this is an individual for whom it's not going to make any difference at all, then we don't feel that it's particularly important to offer it. Next slide, please. So I just want to have a word about mosaicism, because this is something that affects quite commonly people with tuberous sclerosis. And I'm just showing you that lovely picture there. My husband is a gardener, and he wins the Dahlia Cup at the Wandsworth Horticultural Show regularly. I think he's appeared in the last 4 years on the cup, and this is one of his award-winning dahlias, and it just illustrates mosaicism very beautifully. So essentially what this is a hybrid that's meant to have orange and red stripes. These have been bred to have these orange and red striped, but something has happened, because this will be caused by genetic variation. Two strands of basically genetic changes, one causing the red and one causing the orange. And some of these petals have lost the orange component and have reverted back to a red only. And so this is effectively what we call a mosaic, where you've got some petals containing one particular genetic makeup, some other petals containing a different genetic makeup. So if you move on the slide, Luke. Essentially, what mosaicism is when an individual does not have a genetic change in every cell. So in the case of tuberous sclerosis, you might have some cells that have completely healthy Tuberous sclerosis genes, TSC1 and TSC2, don't have any genetic change, but then other cells in the body will have a genetic change. So they're a bit like the dahlia, where they've got different genetic makeups in different cells of the body. Next slide. Sorry, next, if you keep going through. Yeah. So we find mosaicism quite commonly in people with tuberous sclerosis, and this may be what we call somatic variation. And somatic means it's arisen in a particular organ or more than one organ, or it can sometimes just be present in the egg or sperm. So the rest of the body does not have tuberous sclerosis, and that person is not at risk of developing symptoms of tuberous sclerosis, but they are at risk of having a second child with tuberous sclerosis. And this is really hard to determine. Because obviously we can test sperm and many well, some of my patients participated in a research project to look at mosaicism in tuberous sclerosis and other genetic conditions. And we did indeed find that some people had an elevated amount of the tuberous sclerosis gene change in sperm, but not in the rest of their body. But obviously, we cannot test eggs because if you do a test in an egg, that egg is no longer viable. So this is a tricky one, and we get round this if somebody's had a child with tuberous sclerosis and wants to go on and have testing in a future pregnancy, then we would offer them testing. Now in the NHS, we have access to new technology, which is called deep sequencing. So if I go back to my library full of books, and you think of our two books being the TSC1 and TSC2, what we've done normally when we do a genetic test is we read the book forward and we read it backwards to see if we can find the spelling mistake in that alphabet of the A, C, G, and T. However, we've only read it maybe a few times. And if you are mosaic, then that means that you might have the genetic change in maybe 10% of your cells. So reading the book a few times, if you've only got it in 10%, you might not have come across it. You might just not have found it. So we then need to read it 1,000 times to detect those genetic changes that are not present in every cell. And with this new technology, we think we've increased the rate, possibly the pickup to around heading for 95%, but it's still not 100%, even those who are clearly clinically affected. So one of the things that we do is we think about testing an affected tissue such as skin, and I've had quite good success when testing facial angiofibromas. And we've just recently conducted a national audit. I was very, very heavily helped by one of our junior doctors in doing that. And we found that of the people who'd been referred, we had about 55 people referred for the test that we got information on. Over 150 had been referred, so we had about a third of them. We found that of those people, there was a 40% detection rate. Though these are people who previously had no mutation identified. So that was a new genetic result for them. And importantly, if, for example, if we identify this in a newly diagnosed child, the risk that their parents or siblings have tuberous sclerosis as well is very low indeed, because this will be a genetic event that happened after the egg and the sperm got together in the early embryo. And if you have a mosaic genetic change and you're about to have a child, the risk to your future child is going to be difficult to quantify, because although it might be 10% of your cells that we've tested have that genetic change, that might not be the same in your egg or sperm. It might be lower, it might be higher. Next slide. I think this is my final slide. Yes, so conclusions, as I've outlined, there's still quite a lot that we don't understand. In particular, We aren't sure about that 5%, 5 to 10% that we still, the people we still don't find a genetic change. Is that because it's very at very low level, like 0.5% of their cells have the genetic change? It could be that's only confined in one particular organ, so the genetic change has just arisen in the kidneys, and we think that happens quite commonly. Or are there variants in this dark genome where we are beginning to find more and more genetic variation? And we know that that does happen, but we're not very good at looking for it yet, though technology is getting better. The other thing that people often ask me is there a correlation between the type of genetic change you have and the outcome for the individual? So the only situation in which we can predict that is when there's that deletion of PKD1 and TSC2, when we can predict that somebody's going to have tuberous sclerosis and polycystic kidney disease, but we can't predict any more than that, really. So if you look within families who all have the same genetic change, we see a lot of variability. So that is telling us already that there isn't a very close correlation between the type of genetic change and the outcome. So we still have a lot to learn, which means it's interesting for us, but frustrating, I think, for people that have the condition. So I'm going to finish there, and I'm really happy to take any questions.